All right, good morning, everyone. Welcome to the Cantor Global Healthcare Conference. I'm Jennifer Kim, one of the research analysts at Cantor. I'm looking forward to hosting our next panel presentation with Adaptimmune and INAB. I have with us Adrian Rawcliffe, CEO of Adaptimmune, and William Ho, CEO of INAB. Thanks to both of you for joining us. Maybe we can start off. Could you each introduce yourselves and give an overview of your company and its key programs and priorities? And Adrian, we can start with you.
So Adaptimmune is the leading cell therapy company focused on solid tumors with engineered T cells. We are midway through the process of filing our BLA for our first medicine, which will actually be the first engineered T cell in the solid tumor space, which is a huge landmark for the field and for patients with synovial sarcoma. That medicine has completed its pivotal study. We've filed the preclinical and the clinical sections, and we are midway through filing the all-important CMC section due in Q4 this year. Behind that is a broad and deep pipeline of cell therapies, all focused on all cell therapies, all solid tumors, across a broad range of solid tumors. Immediately behind afami-cel for synovial sarcoma, the lead program is a program that we affectionately call CD8, which is an enhanced targeting therapy which is being developed in specifically ovarian, head and neck, and bladder cancers at the moment. And behind that, other well-known targets like PRAME and CD70 complete the portfolio of a wholly owned portfolio of the leading engineered T-cell therapy for solid tumors.
Okay, thanks, Adrian.
Great. Hi, William Ho. I am the CEO and co-founder of INAB. I know many of the people in the audience. I've actually been in biotech for over 22 years. Uniquely, I've been in all sides of the business, covering investment banking, operations in the early aughts, research, and on the buy side. I never expected to become an entrepreneur or CEO, but was actually doing a lot of diligence on the CAR T companies when Kite and Juno came public. Somewhat fortuitously, I came across my chief scientific officer and scientific founder, Dr. Larry Lamb. Turns out he was one of the best experts in the area of gamma delta T cells. Today, INAB is one of the leading companies developing gamma delta T cells for cell therapy. Our goal is ultimately to achieve what we call cancer zero, the safe elimination of all of the tumor cells within the cancer patient's body. And we believe that the gamma delta T cells have the unique properties that allow them to do that. Today, we have 2 ongoing clinical trials, 2 or 3 actually, 2 phase 1 studies. We'll have data this fall, both at the Society of Neuro-Oncology and likely at ASH. And we just opened for enrollment our first phase 2 clinical study in frontline glioblastoma.
Okay, wonderful. Thanks to you both. There's a lot to cover, so I'm looking forward to it. Adrian, maybe we can start with you on Adaptimmune. You talked about the progress you've made on the regulatory front for afami-cel. What's left to do here to get to submission, and walk us through maybe the commercial pathway as well as potential full approval down the line?
So afami-cel, as I said, has completed its pivotal study. 40% response rate, durable responses, median duration of response is 12 months, and that was the basis of the clinical module that we submitted. What remains to do is the writing, review, preparation, publishing, and submission of the CMC module as the last part of this rolling submission. As we've made progress with the FDA over the last couple of years, we have RMAT designation. So we anticipate a priority review. We have RMAT designation, and that's enabled us to have ongoing discussions on many aspects of the CMC and other aspects of the file, including a discussion around taking advantage of the remit of RMAT to enable us to provide alternative confirmatory evidence other than a randomized controlled trial. And we agreed with the agency that the data that we're collecting in cohort 2 of that pivotal trial, which has fully enrolled and dosed patients, and that data will be available probably middle of next year, fully matured. That data can support full approval of afami-cel at some point immediately, shortly after our accelerated approval, which we anticipate being third quarter next year.
Okay. How quickly could it convert into a full approval upon data?
Probably within the year. I think the most important thing from an agency perspective is that the patients have been recruited, that those patients have already been dosed with cells, and that therefore the execution risk associated with the confirmatory evidence is relatively low. And we've already confirmed that the response rates that we've seen in that second cohort are very similar to the response rates that we saw in the first cohort.
Okay, and then can you talk about your commercial strategy and what you're doing on that front?
Certainly. So synovial sarcoma is a rare sarcoma. Almost all sarcomas are rare. Synovial sarcoma is a rare sarcoma. It affects probably something like 1,500 people a year in the United States. And of those, something like 600 would express the right antigen. So this will be a very focused launch with a lot owing to rare disease as well as rare oncology. So we look to how, for example, Immunocore have commercialized their products in uveal melanoma, which is a very similar population size, as to how we think about this. Now, there are important differences. Specifically, synovial sarcoma is treated in major sarcoma centers, tertiary centers, most of which have been involved in our clinical trials. And so we'll be doing a focused commercialization based around treatment centers that largely overlap with our clinical trials and building to probably about 30 centers over the course of a couple of years.
Okay, and can you talk about the recent transition of lete-cel from GSK? How does that play into potentially adding onto your commercial opportunities, and what are you looking to show in data later this year?
Yeah. So lete-cel, which targets another target called NY-ESO, was a product that we developed and then partnered with GSK. And when GSK got out of cell therapy, they very kindly gave it back to us. And they gave it back to us almost all the way through a pivotal study. So GSK is completing that pivotal study as we go through the course early part of this year. And we anticipate getting that product back with data sometime in Q4 this year. And at that point, we'll be able to understand the data and the stage of the product and make a decision on whether it is feasible to continue development and expand the treatment population for that. It is, at the end of the day, an option on a very, very late stage asset. So I think what we look to is, in the event that those data are positive, and we would look to see something that looked similar to the data for afami-cel to be a registerable product. In the event that data is positive, the commercial channel for lete-cel is identical to the commercial channel for afami-cel. It has a broader population targeting both synovial sarcoma and myxoid round cell liposarcoma, another similarly sized population. But the commercial infrastructure and effort required is almost identical to the effort that we'll be putting into afami-cel. Same centers, same doctors, and a very similar patient group.
Okay, and I mean, a lot going on at your company. You also have a next-gen MAGE-A4. Can you talk about where you're at in terms of ongoing programs for that asset? You also have a presentation at ESMO.
Yes.
Would you like to set the stage on what we should expect?
Yes, we're delighted to get an oral presentation at ESMO. I think we've had poster presentations in previous years. And what those poster presentations have demonstrated is that in a large, essentially basket MAGE-A4 expressing trial across all tumor types, the next generation MAGE-A4 targeting T-cell delivers 35 to 40% response rates, and the duration of those responses is increasing. It's now at about 5 months for all of the patients, and these are heavily pretreated patients spanning gastroesophageal cancers through to ovarian cancer, the odd sarcoma patient, and everything in between. So I think that is I think, the best data set for a cell therapy in a broad range of tumors in late-stage patients, and we've decided that we are focusing down on ovarian cancer, bladder cancer, and head and neck cancer, where we're seeing response rates between 40 and 55%, and where there are clear opportunities for development in specific patient populations. Starting with ovarian cancer, where we have initiated a pivotal grade trial designed to assess CD8, the second generation program, on its own as monotherapy and in combination with nivolumab against historic standard of care in the platinum-resistant ovarian cancer setting. And so that trial is ongoing. For head and neck and bladder cancer, we're looking to move into earlier lines of therapy. And so we have initiated cohorts in early line treatment, first, second, third line, in head and neck and in bladder cancer, and those cohorts will read out next year. ESMO specifically this year will be largely an incremental update, validating those signals that I talked about, and with a little bit more follow-up on the patients that we recruited last year. And in addition, we'll have a 10-patient cohort in combination with nivo to primarily establish safety of that combination as we move into thinking about treating with cells and nivo in the earlier lines.
Okay, so the readouts in the second half 2024, that's the highlight. Okay, and then you also have the Gavo-cel program targeting mesothelin in ovarian cancer. What are you looking to see in the more mature phase 1 and 2 data that's also coming later this year?
So Gavo-cel was being developed by TCR Squared, and now by us following the merger in the middle of this year. Gavo-cel targets mesothelin. I think the program is distinguished by, in its monotherapy in the phase 1 study, I think being one of the very few mesothelin targeting cell therapies that actually has a therapeutic index, and was able to be dosed safely and deliver some benefit to patients. However, the response rate, and more precisely the response durability in monotherapy, was insufficient. And so the phase 2 trial that you referred to is a combination of Gavo-cel and nivolumab, and that has been recruiting through the first half of this year. And what we're looking to see is whether we can deliver response rates that are consistent with the ability to develop via accelerated approval against standard of care, historic standard of care. So for example, that would need to be, in our view, at least 30% response rate, and those responses would need to be durable to at least 6 months or so. The patients were recruited first half of this year. We should have readouts on the duration of those responses to that mark in the fourth quarter this year, and we'll be able to make data-driven decisions, which is the key with our portfolio.
Okay, I want to touch on that because there's a lot going on at the company, a lot of changes going on. You've guided to a cash runway into early 2026, and you've talked about making those data-driven portfolio decisions. Could you sort of walk us through what that could look like, what sort of the key points where those decisions will be made, and what's currently embedded into your runway?
Yeah. So I think we have, as you said, a robust portfolio of early stage and late stage opportunities. I think the key is we will only progress into late stage development with assets that we have high conviction in based on real clinical data, and particularly for cell therapy at this point in time, an objective response rate that clearly differentiates from everything else that is out there. And that's the promise of cell therapy. And so that's what we're going to be making decisions on. So we referred to a couple of the decisions that we've got coming up. One is, does the data on lete-cel that we're getting back from GSK in the pivotal study support registration of that drug? Yep. Secondly is, does the data on Gavo-cel support our continued development specifically in ovarian cancer? And then I think there are other decisions probably for next year in terms of the signals in head and neck and in bladder cancer in early lines of therapy as well. And again, we will take very rigorous database decisions based on the product profile, because actually we probably have more things to develop than we could do, and it is reasonable. So we have the luxury, in a way, of picking the best things out of the portfolio to move forward with. And we're and this is in the context of, by the middle of next year, we intend to be a commercial company with first products, and so the stuff that comes behind has to contribute to that as we build that commercial footprint and develop into, I think, the first or only independent engineered cell therapy company.
Okay, thanks, Adrian. Looking forward to see things develop. William, we'll move on to you for INAB. Could you give us a brief overview of the biologic rationale of GDT cells, and why should we be paying more attention to this cell type?
Yeah, look, when I first started the company, everybody was focused on the first generation of CAR Ts that were focused on alpha beta T cells. They're remarkably powerful cells, and they're fairly easy to manufacture in that about 70% of our blood population of the white blood cells are alpha beta T cells. The challenge with the alpha beta T cells has been toxicities. We've seen cytokine release syndrome, we've seen ICANS, all of which have resulted in patient deaths. As the industry has moved towards going towards off-the-shelf therapies, we've moved towards the NK cells, or the natural killer cells. Unfortunately, the early clinical data has demonstrated that their lack of persistence has resulted in a lack of durable responses. We've seen all of those companies get crushed on their stock price. The gamma delta T cells sit at the nexus in between the alpha betas and the NK cells, and so they're somewhat a little bit like both. They have persistence. In fact, Carl June, one of their earliest patients that they had dosed with their CAR Ts, as it turns out, 10 years later, 50% of those cells with CARs are actually gamma delta T cells. But the gamma delta T cells are also not what they call MHC restricted. They don't have to have seen the antigen prior. They can kill directly like an NK cell, and they can use, importantly, they can use the entire repertoire of their receptors to make the distinction between healthy cells and leukemic cells. I think Adrian made a good point about therapeutic index. As we go to these more difficult and more challenging targets, we need to make that distinction, because today the CAR Ts, they don't. They wipe out the entire population of the cells that express the targets. And so in B cells, we can wipe out all of our B cells. As we go to other leukemic targets and we go into the solid tumors, unfortunately, you need those organs. You need your brain, your liver, your lungs, everything else. And so we need to be able to make that distinction.
Okay, can we talk about INB 200, your autologous product? You presented some first-line glioblastoma data at ASCO. What did we see in that data, and what is the unmet need here?
So let's talk about the unmet need first. We're in glioblastoma. The standard of care in glioblastoma has not changed since 2005. And so these patients have really seen little benefit, even with the introduction of checkpoint inhibitors and CAR T. The reality is we believe that the landscape has changed. If you listen to Bob Califf and Pazder, the FDA is now looking for randomized control studies. Single-arm studies looking for response rates in many cases is not acceptable until such point you have a randomized control study enrolled. And so we believe going into indications with significant unmet needs like glioblastoma will help, because there are other pathways in which you can get approval. Going after the 10th DLBCL, follicular lymphoma, or any of these indications, we believe will be challenging. And so at ASCO, we actually had an oral presentation. We were one of 4 CNS tumors to get an oral presentation out of hundreds. It's a phase 1 study, dose escalation. And there, we had demonstrated that we have patients out as long as 2 years without relapse. And so today, we're currently enrolling and completing dosing in our 3rd cohort. We expect to have a presentation at the Society of Neuro-Oncology in mid-November. And we're excited to continue to see longer durability in these patients. Our goal is ultimately to keep them out walking around because tumors like glioblastoma are ultimately a death sentence. The typical progression-free survival is only 4 to 6 months. Typical overall survival is only 14 to 15 months.
And this year, you also initiated a trial to test both auto and allo GDT cells in glioblastoma. You haven't started the allogeneic portion yet. What are the determining factors of whether you end up going the allo or auto route? And when could we expect, I guess, preliminary data from that trial?
So that study is what we call INB 400. We filed the IND, and as I just mentioned, the autologous portion of that study is now open for enrollment. The allogeneic study, as per new guidance late last year from the FDA, requires a separate IND that we hope to file sometime next year. Ultimately, what's going to drive the difference between allo and auto is data. The advantages that everybody knows of an allo product is that it can potentially be off the shelf. We can sell these like our drugs, like the monoclonal antibodies or even small molecules. Manufacture the product, cryopreserve them, store them on the shelf. When a patient comes in, take it off, send it to them, and they can be treated immediately. The other advantage of an allogeneic therapy is that the cells that you manufacture come from a younger individual who doesn't have cancer. And so highly likely that those cells are going to be more potent, more cytotoxic, and have greater killing ability. However, the challenge is that that's not necessarily how your immune system works. Our immune system isn't just a single cell. Potentially, the autologous programs may actually have greater strength because the allo has to have a greater purity. I may have residual NK cells, or I may have some residual alpha beta T cells, and how that mixture of cells interact with each other and cause expansion and additional killing may drive differences in overall survival. And so ultimately, we let the data drive that. And so we'll run a study where we'll actually compare head-to-head in very similar patient populations, both the allo and the auto product in both the frontline and relapsed settings. We believe the FDA is actually very excited about that because I don't think anybody's done that with a single construct to date.
Any thoughts on potential timing for data?
We're just getting enrolled in that, and so our hope is likely in 2025.
Okay. Maybe we could go a little more granular on the update for INB 200 this year. Can you sort of frame what kind of data we should expect, how many patients, the level of follow-up, any color there?
So at ASCO, we presented data where we had dosed 8 patients. Our study in the phase 1 to complete the third cohort will require a total of 6 patients. That would be a total of 13. What we've given guidance is that we will continue to enroll patients, and we expect to complete enrollment this year. If we pull forward and extrapolate from what we presented at ASCO, a number of patients in the third cohort will be in that 6 to 8 month time period, and so we'll see a couple more patients being dosed.
Okay, and I want to move on to INB 100, that's in relapsed refractory AML. You also have some encouraging early data here. Can you talk a little about what we've seen there and anything in particular we should look out for in the update later this year?
Yeah, so I would say it's more than encouraging. I'm actually really excited about this leukemia program. We actually presented April 21st at the European Society of Bone and Marrow Transplantation. At that point, we had announced that we had dosed 7 patients, and every single patient remained in complete remission. The longest remission with a complex AML being over 3 years. And so these patients were very sick patients. Some of them had 3, 4, 5 different cytogenetic abnormalities. We had trisomy 8, deletion 7, P53, FLT3, IDH. Some patients went through 4 prior lines of induction therapies, including Tecartus, just to get to transplant. And so this is in the combination of transplant. Our goal, as you know, much of the challenge in cell therapy has been a lack of durability. Our goal has been how do we design trials and how do we design treatments for the purpose of durability? Because it's how we look at the landscape. As my chief scientific officer often says, look, if you have cancer, you don't care if your tumor shrinks for 6 weeks. What you want is to be walking around for 2 years without progression of that disease. And so we focus our design and our approach for durability. At EBMT, 100% of them remained in durable remissions. As I said, 3 of them having been out all beyond 2 years, 2 of them beyond 3 years. We expect to give an update at ASH later this year. And so we've also said that we would complete the enrollment into this trial. To complete enrollment would be 10 total patients having dosed, and we'll be excited to see the durability of those and how many patients remain in remission. To highlight these programs, we're actually going to have an R&D day. Our R&D day will be October 12th here in New York City. We announced yesterday we have 2 speakers. All of our patients are undergoing the Hopkins protocol. It was designed at Johns Hopkins. The first author on that paper was Leo Lesnick. He'll be speaking at our R&D day, talking about the Hopkins protocol, allotransplant, and GVHD. Our second speaker will be Michael Bishop, Director of Transplantation at the University of Chicago, who also heads the EORTC conference for relapse in leukemias. And so we'll be excited about giving updates this fall.
Okay, looking forward to it. And what is your more broadly thinking plan of attack going forward in AML?
AML is obviously a very difficult indication. I think there's a lot of people who are trying different, targeted therapies, whether they're FLT3, IDH, but really, for the last 40 years, we're still stuck with 7+3. The biggest problem in AML is that people are induced, bridged to transplant, they're transplanted, and they relapse. In the original Hopkins protocol, 50% of them relapse at 1 year. As I said, we had patients out over 3 years. We're going to continue with this trial. We'll have data later this year. But ultimately, we know the goal is to move forward in the induction phase. We have unique CAR-Ts. We announced earlier this year, we have a platform that we call non-signaling CAR that targets CD33. We can also target CD123. The challenge with those targets is that they're on expressed not only on the leukemic blasts, but also in the bone marrow. You may be aware one cell therapy targeting CD33 this summer was put on clinical hold because 2 patients had died. What we're demonstrating with our non-signaling CAR platform is that the gamma delta T cell can make the distinction between the leukemic cell and the healthy cell. Against CD19, we showed 80 to 90% killing against the leukemia and 5 against the healthy B cells. We've showed some preliminary data against CD33 with about 30% killing and about 5. At ASH, we'll give an update on our constructs. We're trying to get higher and higher on the leukemic blasts while leaving the healthy CD34 cells alone to get a therapeutic window. In addition, we're looking at other indications like MDS, where we think there's a path forward, where second line is watchful waiting and not much there.
Okay, wonderful. Thank you, William. I think we are at the end of our time, so I want to thank each of the panelists again for joining us today. And for everyone in the audience, enjoy the rest of the conference.
Thank you.